you had a very interesting experience working with the Dead Sea Scrolls. As a student of history, that like mega history, almost ancient history, that must have been amazing. So just to some background, in 1947, there was a shepherd boy who was looking for a goat of his that he lost, and he threw a stone into a cave. This is in Qumran, which is now present day West Bank, and he heard some jars break open, and in there lay the Dead Sea Scrolls, the oldest manuscripts of the Bible that are available to us now, and perhaps the most important historical manuscript find ever. You had a hand in trying to piece that together using very, advanced biological, we're talking about sequencing, DNA sequencing. So if you can tell us how you got involved in that, and a little bit in regards to what you did to help push along that story.
Sure, absolutely. I'll get to that in a second. I'll just tell you as a segue to this story from the previous story. Yes, I'm very interested in history. In the past, also papers were written very differently. We now have a bigger project where we are trying to replicate. First of all, we are translating and then choosing the most interesting studies. We're trying to replicate work that was done in Vienna in the beginning of the 20th century. In this institute, it was called the BVA.
Yes.
There's no trace of it now. It was destroyed, and everyone working there was either deported or killed. And they published hundreds of papers. Some of them are also dealing with inheritance of acquired traits and questions like that, which interests me. So we are going back to these studies. We got a grant from Human Frontiers to go back and choose and revive these forgotten papers and try to find the most interesting ones and replicating them.
Oh, that's interesting.
This is something I will also involve. Then I will also involve Q&D in that, in trying to assess whether the papers are worth replicating or not. Okay? But yes, I'm interested in many historical things that are related to science and in general. And the Dead Sea Scrolls project came about by total coincidence. So when I just started my lab, that was in 2012. This is literally the month that I joined Tel Aviv University. We had a retreat for all the new faculty in the university. People from all corners of the campus, so arts, science, everything. And then in the evening, they took us to a restaurant, and we took a bus from the hotel to the restaurant. And on the way there, it was a 20-minute ride. I sat next to Noah Mizrahi, who was back then he's a biblical scholar. Now he moved to Jerusalem University. But and we started talking about what I do in the lab and what he does in his studies. I told him about nematodes, C. elegans, epigenetic inheritance. And he told me about the Dead Sea Scrolls in Qumran that he studies from a linguistic angle and so on, and as a biblical scholar. And we started thinking, what can we do together? And then he told me, the thing about the scroll, there are thousands of different pieces, and it's a big challenge assembling them. It's like trying to assemble an unknown number of puzzles with an unknown number of pieces, when many of them are already missing. And he told me that there was a researcher many years ago who tried to look at holes that the worms made in the skulls, because they were lying in caves for 2,000 years, and trying to look at the entry point and the exit point of the worm in an attempt to match fragments that are connected. And then we continued to think about this idea, and we understood that biology, not only of nematodes, but of the fragments themselves, the parchments themselves, could help us. And what we did was we took advantage of the fact that the parchments are written on animal skin.
Yes.
Are made from animal skin. So from each piece, we extracted the ancient DNA of the animal from which it was made 2,000 years ago. And then we tried to match the pieces based on their genetic similarity. We used ancient DNA sequencing. We sequenced literally billions of sequences. It was very deep sequencing. The coverage is extremely low, and the DNA is very fragmented because it's ancient. And we managed in certain situations to piece some fragments together and also to piece apart certain fragments. And together with Noam and other people that gave us other perspectives from an archaeological perspective, a biblical scholar expertise, we managed to make some interesting connections between some scrolls, and also to break some connections that were suggested in the past. It's the most interdisciplinary project that I was part of. I was a part of a few different interesting collaborations, but this was the most fun. By the way, this paper was also 4 years under review. In the end, it was published. Everything was okay, but it was under review for a very long time, and the project itself took more than 8 years, or something like 8 years.
Fascinating. And I think ultimately what you, correct me if I'm wrong, one of the biggest, the ways that you were able to piece these things together is realizing the fact that some of these skins were from cow and some of these skins were from either goat or sheep. And that at the time when these were written, they were essentially, it was impossible to herd cows in that location. So that allowed you to determine where they came from. And then that gave you completely different context as to what was being written by who and the provenance.
Right. So one of the things was really to show that since 2 pieces were found to be derived from cow skin, most of the pieces were made out of sheep, then we understood that they must have come from elsewhere. And this shed light on some weird things in their connections and so on.
Right.
So that was one thing. Another thing that we managed to say regarding provenance, which was very telling, is that, I mean, most of the scrolls that were found in Qumran. This is the main site, which is, these are caves next to the Dead Sea. But some other pieces were found elsewhere, and we sampled pieces from all over the area, including from other areas like Masada. Masada was the last stronghold to where the Jews ran away, and there's this famous story of they were there until they were sieged, and eventually they all killed themselves. And some of the scrolls that were found in Qumran were unknown before, and they are very Qumran specific. It's called Qumran. I forgot now the technical name, but it means that they are very typical of Qumran. Okay. The style and the things that they talked about and so on. One of these documents is called the Songs of the Sabbath Sacrifice, which is really Qumran specific, Qumran typical. Okay. And then, and in general, there's a big question about the Dead Sea Scrolls, which, as you said, are the most important archaeological finding we have in Israel. The question is, yes, we found 25,000 fragments of these scrolls and lots of text that tells us a lot about this crucial period in the history of the area when it's important for Judaism, it's important for Christianity because Jesus might have been there and so on at that time. But the question is, how representative are these texts to what happened to the land in general? Imagine now that the country is destroyed in a nuclear war or something, and all that remains is the library of someone who was hiding in some cave in the desert, in the most difficult place you can imagine to live in, super hot. How representative would his library be compared to the library of a normal person living in Tel Aviv?
Right.
Or perhaps this is a total weirdo and there's no connection between that person's library and what we read in Tel Aviv, right? And we learn about the period based on what was found in Qumran in the desert where it's 40 degrees and there's no water and it's really hard to live. So what was interesting is that this Songs of the Sabbath Sacrifice, which is very Qumran specific, was also found in Metzada, which is the last bit. And people didn't know whether these songs, the fragments, the parchments, were just brought there by people who escaped from Qumran to Masada, because this is the most precious belonging. If you have to run, you take your scrolls with you, right? Or perhaps it's indicative of these songs being more common and well known across the land.
Okay.
If the sources are different, it will tell you that the collection in Qumran is much more important because it's more representative of what happened in the land in general. What we found in this particular case is that the scrolls in Masada and the scrolls in Qumran of the Dead Sea of this particular song, they both derive from sheep, but from 2 different breeds of sheep that are unrelated. All the scrolls in Qumran come from one particular type of sheep, and in Masada, the songs come from a totally different breed of sheep, suggesting that they have that didn't come from the same place. So maybe the scrolls were more, maybe the text in Qumran were more representative of what happens in the greater land in general.
Beautiful. What a beautiful example of interdisciplinary science, or just research in general. What is the current state of the Dead Sea Scroll project? Have they been completely solved, or are they still being worked on?
No, there's still some ambiguities and some places where they don't know exactly whether this piece fits here or there. And we are still involved in the lab in some ancient DNA work together with the Israel Research Antiquity authorities, doing all kinds of crazy things that will, again, probably take years to report. But yes, it's great fun. And this is something that we started our conversation by talking about how scientists are dependent on publications for their promotions, so here, this paper of mine was published in Cell, it was the cover of Cell, but many people told me that it doesn't help my, career necessarily, because it shows that I'm unfocused, or but of course I don't care. but this is one aspect of the architecture of academia, which is very problematic. That you are supposed to focus and just carve your niche. And part of the reason that this is strategic career-wise is that when you work on one particular very defined topic, then over the years, because your career takes 40 years, you know everyone in the field, you know your reviewer. You review their papers, they review your papers, you review their guides. It's much easier to make a career like that. When you suddenly divert to all kinds of weird things, it's harder. But I think this is what we are. But personally, I just need to follow my curiosity. We're not doing it for the money, we're doing it for the fun.
Absolutely.
So I think I think sort of, again, detaching the assessment from the human aspect, which is very limited, would benefit science, in my own opinion.
And to close the story, what an amazing, your next paper you publish perhaps on the Dead Sea Scrolls, you use QED to help you review that, and to go from 250 BC all the way to 2025 using AI to close that loop is quite amazing. I do have one technical question for you. You mentioned coverage of the genetic sequencing. What was you said very little. What was the most coverage that you got outside of a control?
Right. So we had many controls. The controls are actually part of one of the most beautiful aspects of this paper, which I like a lot. I really recommend people to go and read this paper. I think it's my coolest paper.
It's a great paper.
Thank you. But we sequence both and analyze both the nuclear genome of the animals and the mitochondrial genome. We had higher coverage in the mitochondrial genome. And it's much easier looking at the mitochondrial coverage. I don't remember exactly the numbers of the coverage, but it was extremely sparse, which made the bioinformatics and in general the challenge a big challenge.
Yeah, for those who are not familiar with coverage, it's essentially if you take two samples that may or may not be related, if they're related and you sequence their DNA, how much overlap and identical matches there between two sequences. That's what we call coverage. And the more that you get, the more sure you can be in the identity of the sequence and the reliability of the overlap. So it's an important measure. And also, the animals that grew here 2,000 years ago were not exactly identical to the animals that are here now, which was another challenge. Of course, the challenge of lots of contaminations, many of them modern, of humans and other organisms. It was a very challenging project. You need to do all the ancient DNA according to the highest standard, which means you work in clean rooms, in astronaut suits and so on, and sequence very deep. And you distinguish fragmented DNA sequences with ancient DNA patterns from modern sequences in many different ways. Fascinating.